Hello! In today's video, we shall learn in detail about genus Cycas. By the end of this video, you will have a clear understanding about the internal structure of Cycas root, stem, leaf, and also about reproduction and life cycle of Cycas. Let us start with the introduction to gymnosperms. Gymnosperms are a group of vascular, naked-seeded plants in which the seeds are not enclosed within the fruit. Gymnosperms have been classified into 7 orders. 3 have become extinct and the remaining 4 have living representatives. The 2 common orders with living representatives are Cycadales or cycads. Cycas is a typical genus. Coniferales or conifers. Pinus is a typical genus. Cycas ramphi is known as queen sago palm. Now, let us look into the classification of Cycas. Domain, Eukaryota. Kingdom, Plantae. Phylum, Spermatophyta. Subphylum, Gymnospermae. Class, Cycadopsida. Order, Cycadales. Family, Cycadaceae. Genus, Cycas. Species, Cycas revoluta. Now, let us try to understand the morphology of Cycas plant. Cycas is a perennial, evergreen plant belonging to gymnosperms. It consists of unbranched, erect, columnar, stout, palm-like stem with a crown of fern-like pinnate leaves arranged spirally round the apex. A striking feature of the cycad trunk is the investing armor of leaf bases. Genus Cycas is referred to as living fossil because it has not yet altered over the years, while its related members or species have become extinct or fossilized. All the living cycads are strictly dioecious, that is, male and female plants are separate. The main plant body is a sporophyte, diploid, and can be differentiated into root, stem, and leaves. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us compare and learn about the 2 types of roots of Cycas plant, namely Cycas normal root and Cycas coloroid root. In Cycas, the normal roots develop from the radicle of embryo to form taproot system. Coloroid roots develop from the normal root. Normal roots are positively geotropic, that is, they grow downward. Coloroid roots are apogeotropic, that is, they grow upward. Normal roots are less branched. Coloroid roots are dichotomously branched and appear like corals. Normal roots show the presence of root hairs. Root hairs are absent in coloroid roots. Cortex of normal root is small. Cortex of coloroid root is wider, shows 3 zones: outer, middle, and inner. In normal roots, algal zone is absent in the cortex. In coleroid roots, due to the presence of algal zone in the cortex, it is differentiated into outer cortex and inner cortex. In normal roots, the vascular bundle is diarch, that is, there are two strands of xylem and phloem. In coleroid roots, vascular bundle is triarch, that is, there are three strands of xylem and phloem. No infection seen in normal roots. Coleroid roots get infected with algae, bacteria and fungi. Normal roots show secondary growth or thickening. Secondary growth is absent in coleroid roots. Function of normal roots is fixation of plant or anchorage and absorption of water and mineral nutrients. Main function of coleroid roots is nitrogen fixation. The cross section of normal root shows the presence of epiblemma, cortex and central vascular bundle. Epiblemma is the outermost layer of thin-walled cells. Cortex is multi-layered, thin-walled, containing parenchymatous cells which are filled with starch, tannin and mucilage cells. Innermost layer of cortex is the endodermis, followed by pericycle. Vascular tissue shows a central diarch stele showing two patches of protoxylem made up of tracheids. The xylem is exarch, that is, protoxylem is towards the periphery, while the metaxylem is near the center of roots. The pith is either reduced or absent. Secondary growth is seen in roots. Cambium, which are meristematic cells, cut off secondary phloem towards the outer side and secondary xylem towards the inner side. Due to this, the primary phloem is crushed, while the primary xylem is found in the center. The cork cambium phylogen forms cork phylum in the outer region and secondary cortex phyloderm on its inner region. Phylum, phylogen and phyloderm are collectively known as periderm. Let us look at the diagram of coleroid root. The coleroid root of Cycas shows an outer epidermis followed by parenchymatous cortex and an algal zone. The stele shows triarch condition. It is bounded by an endodermis. Next is the transverse section of Cycas stem. Internally, the structure of stem can be differentiated into epidermis, cortex and vascular bundle. Epidermis is the outermost layer comprising of thick-walled parenchymatous cells which are compactly arranged. Cortex is also made up of parenchymatous cells which are filled with large number of starch grains. Mucilage canals are also present. They are lined by secretory cells which secrete mucilage. Internal structure of stem shows vascular bundle which is conjoint, collateral, open That is, xylem and phloem lie together on the same radius, xylem being internal and phloem external, and cambium is present in between the xylem and phloem. Vascular bundle is arranged in the form of a ring and is end arc. That is, protoxylem is situated in the center while the metaxylem is present towards the periphery. Xylem consists of tracheids and xylem parenchyma. Vessels are absent. Phloem contains sieve tubes and phloem parenchyma. Companion cells are absent. In between the vascular bundles are present cells which form the medullary rays and connect the massive parenchymatous pith with the cortex. Pith is rich in starch, sago starch, and has large number of mucilage canals. Secondary growth is seen in the stem of cycas. It is a slow process. A portion of medullary rays which are in line with the cambium become meristematic and form a strip called interfascicular cambium. It forms a ring between the adjacent vascular bundles. Secondary xylem is formed on the inner side, while the secondary phloem is formed on the outer side. Central cambial ring is short-lived, and a new cambial ring is formed every year. Wood formed by this method is known as polyxylic. A well-developed stem of cycas is called monoxylic because the wood is not compact due to well-developed pith, cortex, and broad medullary rays with limited vasculature. Next, we shall look into the transverse section of cycas leaf. Internal structure of cycas leaf can be differentiated into a solen portion and two lateral wings. The wings are curved downward. Cuticle is present which protects the epidermis, followed by a sclerenchymatous hypodermis. Upper epidermis is continuous, while the lower epidermis is interrupted by many sunken stomata. The mesophyll layer in the wings consists of upper palisade cells and lower spongy cells. Transfusion tissue is present around the vascular bundle. They are made up of parenchymatous cells and the walls are not lignified. They contain lignified tracheids with bordered pits. Single vascular bundle is present in the midrib region of the leaflet. Vascular bundle is conjoined, collateral, and open. It is diploxylic. Phloem is arc-shaped and is made up of sieve tubes and phloem parenchyma. Companion cells are absent. Let us look at the diagram. There is a cuticle protecting the epidermis, followed by sclerenchymatous hypodermis. The mesophyll is differentiated into upper elongated palisade cells and lower spongy cells. The diploxylic structure is characteristic of the genus Cycas. There is a thick-walled endodermis bounding the midrib bundle. transfusion tissue is present in the leaf. Let us study the reproduction in cycas. Cycas are dioecious, that is, male and female plants are separate. They do reproduce vegetatively by forming buds on the stem. These buds may be bulbils or adventitious buds which grow into new plants. The male reproductive organ or the male cone is borne on the apex of the stem. It has a central axis and bears a number of spirally arranged microsporophylls. Each microsporophyle represents a stamen. It bears on its undersurface many pollen sacs or microsporangia grouped in sori. Each sorus has groups of 2 to 6 sporangia. In each pollen sac, there are numerous pollen grains or microspores which are haploid. Germination of pollen grain starts in situ, that is, they are still inside the microsporangium. Each pollen grain produces within it a reduced male prothallus, which consists of a prothallus cell, a generative cell, and a tube cell. Further development of pollen grain takes place after pollination. Next is the female reproductive organ. The female reproductive organs are the megasporophylls or carpels produced in acropetal succession above each crown of foliage and scaly leaves. Carpel shows 3 distinct regions: upper leafy portion, middle ovule bearing portion, and lower stalk. Ovules are formed on the lateral side of the middle portion. Ovules are sessile and are borne laterally on the stalk. Next, let us study the structure of ovule. Ovules have only one integument, unitegmic, and are orthotropous, short and straight. The ovules show the following parts: a 3-layered thick integument, an opening called the micropyle, a pollen chamber, a nucellus fused with the integument, a female prothallus, a few archegonia, and an archegonial chamber. Each archegonia has a large egg cell with a prominent egg nucleus in it. Next is pollination. Pollen grains are carried by the wind. They fall on the micropyle, which contains mucilage. When the mucilage dries up, the pollen grains reach the pollen chamber. The tube cell forms the pollen tube, which penetrates into the nucellus. The generative cell divides into the stalk cell, which is sterile, and into a body cell. The body cell divides and forms 2 multiciliate male gametes or spermatozoids. The pollen tube bursts and the spermatozoids enter the archegonium. Next is fertilization. One male gamete fuses with the egg nucleus. This is known as fertilization. The oospore is the first cell of the sporophyte. It is diploid and has a dense cytoplasm and a large nucleus. It enlarges and gives rise to the embryo. Ovule is transformed into seed. The nucellus and the inner layer of the integument are used as nourishment for the developing embryo. Let us learn about the seed. The testa or seed coat are formed by the integument. It has an outer fleshy layer and an inner stony layer. Micropyle is the small opening at the top of the seed. Inner to the seed coat is the endosperm that stores a large amount of food material. The mature seed bears only one embryo with two cotyledons. Let us understand the germination of seed. Under suitable condition, the seed coat swells and breaks as water is absorbed. The coleorhiza protrudes out and grows into the radicle, forming the root system. The cotyledons absorb food from the endosperm and is responsible for bringing the plumule out of the seed coat. The plumule first forms few scaly leaves and then the foliage leaves. Finally, let us learn about the life cycle of cycas. Cycas plant is dioecious. The male plant bears male cone. The cone has many microsporophylls which have microsporangia or pollen sac. The microspore mother cell, which is diploid, undergoes meiosis or reduction division, forming haploid microspores or pollen grains. The male gametophyte produces male gametes. The female plant bears megasporophylls which produce ovules. The megasporangium gives rise to the megaspore mother cell which is diploid. It undergoes reduction division to form haploid megaspore. The female gametophyte has archegonium which produces the egg. The male gamete fuses with the egg to form diploid zygote or oospore. It gives rise to the embryo. From the embryo develops the seed, which during favorable condition germinates into a new plant. So today we have learnt about cycas in detail. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.